I think what I want to share with you is more kind of a way of thinking about origins of life, where individuality is an important part, because the way I conceive of the process of origins of life starts with protocells and ends with cells. So there's a strong sense of individuality, as you will see in my talk, and I will try to make let's say rational arguments to kind of claim for that, and actually retrieve the maybe conventional idea that, you know, something happens within cells that is not happening outside cells, and we have to account for that. And this is going to be important also for origins, right? Nevertheless, even if we have those individuals that are transforming through the process of origins of life, I think it's very important to take into account that within the process of biogenesis, there is a collective effort. The ecological and evolutionary dynamics, which brings you to the collective dimension of the living, is also important. So in the transformation of the individuals, there is a kind of a collective effort that I'm going to try to address in the last part. So, so these are the heroes of my story, bacteria, and just, you know, kind of closing up on what happens within these systems that are, you know, the basis of all biology. First of all, I suppose that lots of you are already aware of this, but they're not just bags. They are very, very complex physical-chemical systems where water is in different conditions. You can have, you know, this heterogeneity in which there are some areas of the cell where you have more kind of gel-like phases, and then there are some others where it's more like aqueous solution, where metabolism happens. So there's kind of a part of it where you can treat it as a pool, but there are some other areas where you have a lot of crowding, and the nucleoid and all on some other areas are so heavily crowded that you cannot think about it as simply a bag. And the complexity is absolutely amazing. The types of structures, macromolecular structures, supramolecular structures, and interactions, including, of course, the membrane, and I'm going to be focusing on that for when I introduce protocellularity, and the interaction with the environment is fundamental to keep the system going, right? So underlying all of that, and in this, especially in these kind of pools that we have here, we have all the, you know, metabolic complexity that maybe you were speaking about the first day. Of course, in particular organisms, you have a subset. This is the metabolic chart, which is the extension of all that we know about metabolism. But particular organisms, in this case, E. coli, carry out subparts of that whole chart. But in any case, incredibly complex reaction network. And so I think that we shouldn't forget that these kind of basic facts from biology, that this complexity is generated within cells, right? So the synthesis of the key compounds that rule the behavior of the system take place within the cells. That's kind of a fundamental fact. And the second, I think, fundamental fact of biology is that all extant biological systems are a result of a complex and very long evolutionary process, okay, starting from the last universal common ancestor. And even if we have kind of difficulties to kind of draw trees, evolutionary trees, phylogenetic trees, this is also a fact that is fundamental, right? So there's a kind of two dimensions or two kind of basic properties that I used long ago during my PhD to try to come up with a definition of minimal life. One is this capacity for self-construction, building your own components. So it's very close to the idea of metabolism, but from a point of view of the theory of autonomy. And the other one is the open-ended evolution, which is related to, as a proxy, you can think about it, Darwinian evolution, but it's a kind of the potential for indefinite growth of complexity, right? With these two properties, it's, of course, there are properties to explore. It's not like I want to encapsulate life and say this is it. It's just to kind of give you a heuristic, and a heuristics that is a systems type of heuristics, in the sense that those properties are not reducible, and they relate to system properties, right? And so this, if you define life in these terms, then origins of life. The idea here is that we're going to have a platform in which you are not thinking just about one type of component, one type of process, but you're going to be thinking in terms of this, what we call, in this review that we wrote, a decade ago, time goes. So prebiotic systems chemistry, so the field of systems chemistry, in which we were proposing that the platform to actually tackle the problem of origin should be with combinations of molecules in different types of processes, right? A combination of processes, and that's why, because it's a combination of processes and combination of different types of precursors, functional precursors of biological systems, then it's difficult to reduce to one single type of molecule process. So the idea here is to simplify, but simplify without going too simple, right? And that's the trick, and it's not easy, right? The idea here is that with this definition of life in terms of autonomy and open-ended evolution, we can also structure like a line of work, a line of research in origins of life, where we start with autonomy in a kind of a basic sense, which would be a kind of minimal metabolism that I'm going to be speaking about, and then develop into different stages where that type of system becomes increasingly complex, right? Developing different types of mechanisms. Some of them could be hereditary mechanisms, some of them regulatory. I will just speak about them very briefly, and we can discuss. But the idea here is that we're going to be using the idea of metabolism as a bridge. So it's not going to be the chemistry of life. Eventually it will. Like the outcome of it will be like what is supporting living organisms, right? But if you want to use it as a bridge, you have to define it independently from life. Otherwise, you're caught in the trap of, saying what is life, right? And life, I think, is not going to be full-fledged until we do the whole process of origins of life, right? So we need a kind of a conception of metabolism that simplifies the chemistry of life, right? But at the same time helps us bridge the gap, right? And in this process, we can have a kind of a reduction of the potential chemical diversity that organic chemistry can provide, but then there is an increase in functional diversity and autonomous control, and an increase in the complexity of the molecules and the organization involved, right? So, so the idea here, and I want to focus quite a bit on this, is that metabolism is not just the network of reactions, be them autocatalytic or cyclic or linear or a combination of all of those, but that's chemical. That's a chemical network, right? For me, for us, the idea of metabolism is a more kind of constructive, like integrative process in which those chemical processes synthesize the constraints under which the whole system runs, right? So it's a kind of a production, but also a control relationship. So it's a kind of a functional bootstrapping, because what you have typically is you have the result of what is the result of the metabolic dynamics that we see in cells is, you know, the enzymes that run the actual pathways, right? All the not only the enzymes are constraints, but there you also have the synthesis of your boundary, right? And control, spatial control of what's going on there, right? So for us, even if we can, the idea here is to kind of be able to simplify and not just try to come so quickly into the, let's say, the metabolic pathways, the biochemistry, metabolic pathways of life, but have this kind of general conception of having a system where this kind of the processes, the chemical processes generate those molecular structures that take control, right? And what is this control? That's the tricky bit, to kind of from biology try to select which are the fundamental controls. And in a paper that I wrote together with Carlos Briones and some other colleagues, we were kind of proposing that it's very difficult to think about an organization of processes where you synthesize and you control. And also from a physicist's point of view, it's very difficult to kind of think about how to do that without some sort of spatial control, some sort of kinetic control, because there are all these processes, chemistry, happens at its own rate, right? But you need to coordinate in space and time the reactions happening, right? So kinetic controllers or catalysis is fundamental, but also spatial control and some sort of energetic control as well, right? Because again, you're going to have to synthesize. It's going to be a thermodynamic effort involved, a very strong thermodynamic effort involved. So you need to, make use of the resources. And you see in biology, actually, I think there is there's a slide here. I don't know if I have it. No, well, okay. Yeah, I thought I had another slide here, but this is enough. So in biology, we have these transduction mechanisms in which, biological systems do not use the energy as they find it in the environment. They actually change it, they transduce it to their own means. Okay. So there's a way of thinking about it in very abstract terms, like work constraint cycles. Okay. So this is an idea from Kaufman that I also took and developed in my PhD. But this idea of having synthesis and control is having flows of matter and energy, right? It can be used constructively for the system, so that's work, useful energy, right? And work itself needs to be constrained. So there's a kind of a loop between constraints and work. Okay, and so I think about metabolism in this way. Okay, you have the laws of physics and chemistry, but then material organization, if it's articulated in the right way, can kind of have its own rules. That's the idea of autonomy. Okay, autonomy is self-rule, but it's not that we are thinking about rules coming from space, but it's just the material, output of the metabolic network that kind of, so it's kind of that functional loop, bootstrapping loop that is critical. And I'm going to be focusing, because that's kind of also connects with the idea of individuality, okay, on compartments, right? Because these compartments it's not that they are separating the system from the world, but they are actually connecting it to the world, right? We wouldn't, the constitutive dynamics that is taking place there is, it's an open system because it's far from equilibrium dynamics, and you really need to be able to actually generate those compartments to have that interface under your control. Okay, that's the idea. Compartments are not just separating. Boundaries are not just separating, like connecting, right? So we start with this kind of a scenario where you don't have just water, but you have probably compartment. This is just oleic acid, but you can think about compartmentalization as a kind of relatively easy process to happen. You have in prebiotic times, I think maybe we have here, yeah. So the basic ingredients are there available. Like you have amphiphilic molecules and you have amino acids for sure. If you can take something for granted, is that these kind of components are available, right? And in fact, if you don't start from, imagine that you don't start from this, you start from a network that doesn't have compartmentalization. The problem is that if you let it evolve and things run, like through, imagine you develop some catalyst, you let it run, then compartmentalization is going to be a nightmare because it's going to suffocate the system, and you actually need to have this kind of coevolution between chemistry and self-assembly of the compartment to take the compartment from these quasi-equilibrium states to far from equilibrium states and host those types of reactions, right? So I've been preaching this for quite a while now. It's much better maybe to just use a quote from a Nobel Prize who actually realized this through an excursion that he did. He's an RNA world guy, him, all of you, Jack Szostak. And in a review about problems related to RNA replication, non-enzymatic RNA replication, has this beautiful quote that all the protocellular structure poses more problems initially, and also mostly to the chemists, right? It's actually simpler to solve these problems up front rather than leave them till later when they could become completely intractable. In fact, if we have some evidence that if you have reaction systems that go slightly more rapidly than just in the thermodynamic kind of regime through catalysis, you're going to suffocate the system, right? So maybe some of you know Yutechu. He was a researcher here at ELSI. Yeah, he sent me. So this is a kind of an elaborate way of saying it, a more direct way of saying it. There's no vesicle, no life. He sent me this T-shirt, and I love it. So absolutely. So we are thinking about simple chemistry coupled to self-assembly. It could be chemistry happening initially outside. It could be chemistry happening at the actual interface. It could be chemistry happening within the compartment. But in any case, this is a kind of the context where you start establishing functional relationships. By functional here, I mean combination of these different components, different processes contributing to the self-maintenance of the system. So it's a kind of an organizational, a physiological way of thinking about function, not so much evolutionary. But evolution is going to come to the picture soon. But you need some sort of phenotypic space, initial phenotypic space, where natural selection can get started. Otherwise, you fall into a too simple. Like if you just have replicate, for example, the classical one is just having self-replication, right? And you basically you go to a dead end because you don't have a phenotypic space that is large enough for evolution. Is that the first one? Oh my God. Okay. Okay, I will run. So we've done experiments on this kind of a scenario. We've done modeling. I was very happy to see that among the posters and among the research that is being carried out here at ELSI, quite a few of you are taking these ideas of having a fatty acid compartment combined with a kind of a simple oligopeptide, having a kind of a let's say cooperative synergy between, you know, what is a compartment, and it could be, you know, a compartment that could be facing an osmotic crisis. Maybe these kind of channels could be an alternative way of releasing the waste or whatever that is compromising the is making it difficult for it to stay. So there's kind of a synergy between different types of components, and this is the idea of function in a very let's say first, like it's the beginnings of functional relationships in biology, right? And together with function and the functional integration of the components, we have to be aware about this coupling with the environment, right? And so the idea of agency that I use here is just simply the fact that through building your boundary, you're going to be able to modulate the coupling with the environment, right? Initially, probably we cannot have these, you know, sophisticated transducing mechanisms, of course, like we have now. But we have to think about, in the same way as we have to think about how enzyme catalysis could have a kind of could be developed in some way. We have to think about how these coupling mechanisms were developed from the initial stages, right? And I think being a system that initially had to be far from equilibrium and coupled to the environment, both in energetic and material terms, make this kind of agency. I know, but for a lot of people, agency is a heavy loaded word, but for me, it's just simply some control on the way you define, so the way the system defines the relationship with the environment, full stop, right? And this is an asymmetry that is important to take into account. So from this basic kind of ground that we are still exploring, and the field of origins of life and protocell camp is still trying to find which is the right combination of components that could give you these initial features. What I want to share with you in the last, 5 minutes? No, 2. How many do I have? Yeah? 5. Okay, 5 minutes. I wanna tell you a bit the story that we are thinking about in terms of how that can develop into different stages, right? So the idea here is that you have an individual that is relatively simple here. As I said, there is function, there is diversity, but it's relatively simple to something that we can call a biological individual, where the relationship between the parts and the architecture and the combination of components that you have there is just amazing. It's just, you know, it's unbelievably complex, right? So you cannot think about the process as a protocell, you know, bringing forth another protocell and another protocell and another protocell. You have to jump to a different level, right? The level of population. So you have the organizational or the individual kind of dimension. Again, this is the different stages of autonomy, right? But you need to also combine that with an evolutionary picture in which you have a population of protocells that initially maybe were not very reliable in their reproduction, but they became increasingly reliable through reproductively through mechanisms of heredity, and this is a bottleneck also in the field of origins of life, reproduction. And so this is not, we are not free of difficulties here, but the idea is this, right? And for example, we are exploring now how could you have regulatory behavior, which also comes from trials and errors that happen at the level of the population and how the population kind of learns from there, right? So we are thinking about, well, by the way, we're thinking about regulation not just as feedback, but as something like a constraint operating on a constraint, like a meta constraint. This is the constitutive kind of metabolic level, and then you would have another mechanism that is generated by the system that, depending on the input that you get from the interaction with the environment, depending on the signal, with something you can call a signal, because it's going to change, it's going to shift the type of metabolism that is going on, right? So we are thinking about regulation as a meta constraint, okay? And that applies to systems that could be like protocell. So let's just jump very quickly to describe the type of a scenario that we are exploring with this new simulation platform, where we have protocell populations, where it could be, this is a kind of a different biomasses. This is a consumer resource model where you can have cross-feeding, you can have ecological relationships between the different types of protocells. And then you insert that into an evolutionary algorithm that takes place at a much longer time scale. So it's a kind of a simulation platform. We are putting together things that are happening at the level of physiology, let's say protocell physiology, relatively rapid changes here in the enzymes or the kind of the transformers of nutrients into different type of outputs here that can be nutrients for other types of species, protocell species here, and you follow the kind of the phylogenetic tree of this kind of species that you're having here, and you have evolution running, but you also have the possibility of a regulatory mechanism here that allows the system to learn from, to can have a kind of a memory. And this regulatory mechanism is going to be changing the rates at which you have these types of enzymes here are kind of assimilating the nutrients, and depending on the forcing that you get from the environment, right? Whether it's a slow forcing and whether kind of phylogenetic adaptivity can happen even if there is a lag here or not. When you go and have more stringent speed forcing that we call it, then phylogenetic adaptivity is not able to cope. And whenever you have this regulatory mechanism here operating, you can have changes in somatic behavior that are kind of adaptations that are not, you don't have to wait for evolution to happen. You can actually learn and implement regulatory mechanisms from the exploration that the population does. We are not putting any fitness function here, just the outflow of the chemical reaction. We let the simulation algorithm run, and the system just learns how from experience, right? Anyway, so just to wrap up and have some room for discussion, the idea here is that through these stages, well, I can get a bit more provocative here, and maybe go a bit and say that there's information. So here you see these concepts that we have here: function, agency, regulation, information. They are, for me, fundamental concepts to kind of relational concepts. Like, we naturalize these concepts. These are concepts that come typically from our human interaction. We have naturalized them in biology, right? But they have their in my scheme, they have their place, right? Where they play a fundamental role in these transitions. And for me, information is absolutely necessary. Some people put it down here, but for me, it's absolutely necessary to kind of try to understand how these populations that we have here, through these stages where they develop autonomy, initially in a very basic way, but already here with natural selection probably operating, having families and clans of protocells, developing ecologies, ecological relationships. But the the way all the way to the last universal common ancestor, I think that what is necessary also for open-ended evolution, which is this capacity to maintain your level of complexity and potentially increase it, you need these genetic mechanisms, right? So this would be the end of the process for me of origins of life, in which you would have, this intuition from John von Neumann, in which you have a universal constructor operating here, right? So below this level of complexity, probably there is degeneration, as he famously, wrote in this quote. And basically, you need to have a kind of a decoupled description, even if it's just a partial description of the system or some of the features of the system, decoupled from the actual metabolic, all the constructor part, to actually keep it and solve two big problems at the same time, which is, one is to have an individual that is very effective in its metabolism and its way of coping with the environment, and you have the description of those components, enzymes, and other types of components that you need to keep the system here and now fighting for survival, so to speak. And at the same time, you have the evolutionary dimension, right? Like this capacity to maintain across generations, right? So I think that the invention of the genetic code and the phenotype-genotype decoupling happens in the late stages of the process is fundamental for the long-term sustainability of the phenomenon. And with that, and with maybe an entry to, if you want to check this dialectical systems entry, where I actually make the case that there's a kind of, so there's the part that has to do with the individuality in the process, but then there is a kind of, there has to be like a collective effort in order to get to these stages, right? So the collective dimension of life happens and it unfolds in the actual process of origins of life, right? And this has an ecological and an evolutionary part, right? So with that, and with maybe a quote, I mean, this is a classical quote, but it's very pointed here from Master Morovitz. Sustained life is a property of an ecological system, and I would say in evolution, I would add, rather than a single organism of a species, with an acknowledgement to my PhD students who are the main source of motivation. I just thank you for your attention. I'm ready to take questions. Thank you.